Hello guys and welcome back to Excelsior Academy. So guys, today we'll be doing homologous recombination. Okay, so this is part of the genetics also, and this we are going to use in order to understand like how the DNA repair is also happening. Okay, so this is also part of DNA repair. Okay, so homologous recombination. So what happens in the homologous recombination? So it is the exchange of the segments, okay, so genetic material between the homologous gene sequences of the chromosome. So you know, and this happens during the meiosis. ये हमने पढ़ा है छोटे में, right? So between the homologous chromosome, so the sister chromatids, okay, so either they are going to have the recombination over there, right? So this also we'll study in detail when we'll do the cell biology, but I am sure that everyone knows about the meiosis and the crossing over happens in the packaging, right? Now, in prokaryotes, it occurs during the conjugation, transformation and transaction, जहाँ पे genetic material जो होता है वो exchange होता है, ठीक है? So already the example of conjugation we have seen in the case of F plus and F minus plasmid. Do you remember, guys? This was already taken when we were talking about different modes of replication, right? Now, in the transformation also, you know, when certain amount of heat shock is given, or sometimes you use certain chemical reagents which will make the cell wall permeable for the genetic material, then the transformation is happening. So again, what does it mean? It means from the outside environment, the cell is taking up the DNA. So again, there is a exchange of your genetic material, okay? Then transaction with the help of the viruses. So in all the scenario, what we see is that there is exchange of genetic material. Okay. Now, there are different models for homologous recombination, of which first one we are going to discuss is about the holiday model. Okay, so of course this name, I mean, it's not for the holiday, but this is the name is given by the Robin Holiday. Okay, this was a guy who actually found for the first time in 1964. Okay, so that's why it is known as holiday model. Okay, so it is also known as heteroduplex model because this model also explains the recombination between short homologous sequences between the two heteroduplexes. Are you able to follow? This sentence is very important to understand, okay? It is also known as heteroduplex, okay? Why? Because the recombination is happening between the heteroduplex, two heteroduplex of the homologous sequences, it is happening. Now, how it is formed, we'll see one by one with the figure. So first two homologous DNA molecules are need to get aligned with each other and the nick is introduced in both double-stranded DNA at the same position. ठीक है? तो सबसे पहले हमने क्या बोला? That the homologous DNA, it should be homologous chromosome, they should be aligned like this. ठीक है? Then a nick should be made at the same position in the two double-stranded DNA position. Then the strand which is next to the nick get unpaired to its complementary strand and free to invade the near aligned double-stranded DNA at the same location where the nick is formed. इसका क्या मतलब है? This strand is going to invade this position and this strand is going to invade this. So this is known as strand invasion. ठीक है? थोड़ा सा idea आ रहा है? If you have seen the figure like the PDF which I have shown, then you will get clear idea. So after invasion, it base pairs with the homologous region within the double-stranded DNA and it forms a holiday junction. And this step, okay, this procedure, this step is known as the strand invasion. Okay, let me show you the figure. Okay, so this is a different figure I have taken because I felt that this is more, more clear than the PDF which I have shared with you guys. Okay, so are you able to follow, right? Shruti, are you able to follow?
Yes, ma'am. Yeah.
Okay, we just have started only and I'll be taking this topic today. Okay, so what is happening? So as I said, two homologous chromosomes, they need to align side by side. So, okay, we have aligned and you can see that capital B, capital A, small b. What does it all represents? It represents they are gene alleles. Okay, so they are alleles. So homologous chromosomes are there. Now, what is happening is that a nick should be formed at the same position. Can you see? So the side by side जो sister chromatid है उसमें आपका nick होगा first. Then a free end of each broken strand migrates to the other DNA molecule. Can you see? It starts migrating like this, and this will start migrating like that. And now what is happening? It's a single-stranded DNA break is there. Okay. So if suppose gap was there, in few cases what happens is that when this nick is made, certain nucleotides are chopped off. Okay. इसका मतलब क्या? They have been removed. So a gap will be present. So we know that whenever there is a single strand or double strand gap is there, your DNA pol 1 will come and start synthesizing the DNA, and ligase will come and seal the DNA. Okay. So you can see over here, the strand invasion starts and they get sealed to the respective opposite sister chromatids. Okay. So each invading strand joins to the broken end of the other DNA molecule, creating a holiday junction, which begins to displace the original complementary strand. Can you see? अब यहाँ पे क्या हो गया? So this entire thing is known as the holiday junction. Now, then the person Holiday, he also suggested that it depends the alignment, how this junction is being formed. It is going to have two products. Okay. Either it is called the recombinant products or the non-recombinant products, crossover products or non-crossover products. We have the other words also. They are also known as splice products or they are also known as patch products. ठीक है? ये सारे अलग-अलग नाम हैं, and but it means all the same. Okay, is this much clear to everyone? अभी हमने सिर्फ holiday junction तक गए हैं. Okay, now branch migration. So it doesn't stop till there only. It is going to migrate. And now this is happening. This migration is only possible. See, इसमें क्या होगा? Sorry. Of course, it is going to break the hydrogen bonds between these, and it is going to form the hydrogen bond between these. Now, why it is possible? Because they are homologous chromosome, and hence they are sharing the complementary base pair. Correct? Is this much clear? Why the branch migration was possible? Yes? Tanvi, clear hai?
Yes, ma'am.
Okay, okay. So, this is how it is going to migrate. Okay. Okay. Now, it is going to get rotated. So, this forms the branch point. Now, look at over here. First, it was straight. And look at, see this figure. हमने कैसे दिया? Red and red blue है, then blue red and पूरा blue है. Okay, ऐसे थोड़ा याद रख लो. Now look at over here how they have got open up. So they got oriented. Correct? Are you able to imagine this thing? हो रहा है? Imagine हो रहा है? Yeah. Okay. Clear? Now again. So now we have got two options. What are those two options? So after this step, holiday junction gets cleaved, and this step is known as resolution of holiday junction. यानी कि क्या? मुझे इसको अभी solve करना है. We can't have ऐसे चिपके हुए two sister chromatids, right? We want to release them out. So there are two methods by which it can be done. That is, either you can have cut in this direction. This is your first side, okay, or you can have cut in this direction. Okay, now let's go one by one. So if suppose you have first this vertical plane wala cut, so what is going to happen then? Kya kya join hoga? This is going to remain as it is. Can you see this much red and blue? This is going to remain as it is. Okay, so we have red and we have got blue like this. Okay, next what we have is red itna sa and blue, sorry, blue over here. Okay, are you able to follow, guys? This one, when the cut is made, I'm gonna get this red which gets rejoined to this much portion of the blue. So I'm getting this strand. So this is one product I'm getting. Clear?
Yes, ma'am.
Okay.
Yes, ma'am.
Similarly, over here, I'm going to have blue. Okay, and ये छोटा सा रहेगा, and then I'm gonna have, see, red, and this much stretch of red. Okay, so let's see this also. So this is no problem. So this is the stretch you have over here. Okay, red lamba sa, and then chota blue. Then you have got blue and then red. Okay. Now looking at these two products, okay, this is the second product. What do you think? Are they similar to the parental? Parental क्या थे? Right? Are they similar to parental? Any of the strand is similar to parental? What do you think? This is one product. This is another product. Do you guys think that it is similar to the parental? No,
No.
right? Because none of the strand is having the parental copy. Both here also, both the strands are having half of each, right? So that's why it is known as the crossover product. Okay, like in few textbook it will be known as crossover product or recombinant products or splice product. Okay, so these are the different synonyms you can have for this condition. Whenever you have such recombinants, okay, so you can see that the crossover product is totally different from the parents. It means it is 100% recombinant, correct? So the scientists, like the genetists, they will understand that the holiday junction, in that the cleavage of the DNA has happened in the vertical plane. Abhi theek hai? So looking at the recombinance percentage, you can tell how much, in what direction the holiday junction has been resolved. Are you getting the point? Okay, so this is how, looking at the percentage of the recombinants, you can tell whether the holiday junction was resolved either in the vertical plane or in the horizontal plane. Let's see horizontal plane also. See, you can see in figure here itself. So if I make a cut like this, okay, so I'm going to have complete blue strand over here and another one with this much blue and this much red and blue. Okay, so blue, sorry, so I'm gonna have blue, then blue, and over here blue, and then red. So this is one product. Okay, when I make a cut over here. Now next I'm gonna have complete red. Okay, complete red, then red over here and this red over here and blue. Now, so here you can say that 50% of the parental copy is present. Yes, because your this strand and this strand is completely of parental. Correct? So, and if you see here, you know, ek chota blue, ek chota red, ek chota blue hai, and because of this, it is also known as patch. Okay, able to follow, guys? Now look it over here.
Yes, ma'am.
So if I had vertical cleavage of the double-stranded DNA, I'm gonna get, sorry, ulta bolte yahan pe. If I do vertical cleavage, then I'm gonna get the crossover recombinants. Okay. If I do horizontal cleavage, then I'm gonna get non-crossover recombinants. So where you can see the parental strands are also there. So the holiday model predicts non-crossover or crossover recombinant DNA depending on whether the cleavage is in the horizontal or the vertical plane. Is it clear, guys? Clear hai ye? Holiday model? Yeah. So इसमें base question कैसे आता है? They will give you certain percentage of the gene allele कि ऐसा है recombinants, like this much percentage is recombinant, this much percentages of the non-recombinants. Then you have to guess whether it is the cleavage of the holiday junction at the horizontal plane or in the vertical plane. So if you could remember this much, the figure and all, then it is easy. Okay. And हॉलिडे मॉडल समझ में आ गया है तो आगे जो हम मॉडल पढ़ने वाले हैं वो और इजी हो जाएगा. ठीक है. No, this is a video which I have got from YouTube itself. This is the link. Okay, so let's listen to this. Okay, very beautifully they have shown how the the homologous strands are getting rotated, and the rotation is very important because हम 2D में दिखा नहीं पाते हैं. Okay, so this video they have shown very nicely. I'm just gonna play for you guys. Listen to that also. Okay.
Understanding homologous recombination by using holiday model, at first we are taking here two homologous double-stranded DNA molecules aligned with each other, which have different alleles of the same gene. In holiday model, at first, on both the DNA duplexes, single-stranded nicks are formed at the same position. Then, the strand invasion occurs. In this step of homologous recombination, initial short regions of base pairing form between the two recombining DNA duplex near the nicks, and a holiday junction is formed. It is the region where two DNA duplexes form a cross-like structure of DNA strands. On the other hand, each of the nicked strand can easily base pairs with the uncleaved strands of the opposite duplex because of having the same or almost similar sequence on both the duplexes. Now, the branch migration occurs, in which the holiday junction moves by melting and forming the base pairs repeatedly. In the parental DNA molecules, base pairs are broken, and in the recombination intermediate, identical base pairs are formed. The DNA duplexes have very small sequence differences as they have the different alleles of the same gene. So, after branch migration, the new DNA duplex will have a few sequence mismatches at those regions. These regions are named as heteroduplex DNA. At the last step of DNA recombination, the holiday junction is cleaved, regenerating two separate DNA duplexes. This process is called resolution. For better understanding, here the holiday structure is being rotated to have a plain structure without crossing strands. Resolution has two possible sides. Each side can give different outcome after cleaving. The first cut site can be the pair of strands which were uncut during the first step of recombination, and having single color, they can be easily identified. As a result, the original DNA duplexes are spliced together in the resulting duplexes, so they are named as spliced recombination product. The resulting duplex will have a region of one pair of identical alleles from one parent, a region having heteroduplex DNA and a pair of alleles from other parent. In the last region, the C allele have switched from one duplex to another. So this is called the crossover region, and thus the product is also called the crossover product. The second cut site can be the pair of strands which were nicked during the first step, and they have mixed colors. As a result, the product have a patch of hybrid DNA, so they are called patch recombination product. The product is also called non-crossover product, as the A and C alleles have not changed their position in the new product and remains identical like the parental duplex.
Okay. Yes. Any doubts? I hope the video was useful, like in getting the in clearing your imagination as well. Yeah.
Ma'am, one question.
Yes. One second. हाँ बोलो.
When you said that there are two nicks formed at the same site on both the chromosomes, how are they formed?
See, whenever recombination is required, then the nick will be formed. We have already discussed, na, endonucleases are there, which results in the nick formation. If you remember, when we were talking about the repair, okay, when we were doing the repair, so we said, na, agar yahan pe damage hai, okay, so you just don't, if you remember in the NER also, we just don't remove this entire, this much strand is removed, right? So we make a nick. This is done by the endonuclease, है ना? So all
but there is no problem over there. There is no reason for the nick to form in this particular case because
that
the
is a
DNA is fine,
requirement.
right?
No, it's a requirement. ये synapse होता है meiosis के time पे. Why it is happening? It's a requirement so that the coming offspring जो भी आपका जो cell है या जो भी आपका बन रहा है, it has got the genetic diversity. Genetic diversity is the need of evolution. Okay. हाँ. Now, I mean, is it clear now why this nick was there and why this is recombination is happening? It is a requirement of the, to become.
Why was clear already, the genetic diversity part. It's just that if the nucleus forms a nick, even though there was no requirement to form the nick. As in evolutionarily, yes, but then technically there is no problem over there.
Okay. Okay. So now if that is clear, then these are the other models. अब कैसे होता है? Why when you say everything ना model, इसका मतलब क्या होता है? It has not been seen, from the what to say? It's not seen directly. It is just guessed by looking at the electron micrograph or other chromosome karyotypes and all. Okay, ये सब देख के हमने guess किया है. Like of course over here is the measles and the radding. So they have understood like this कि ऐसा हुआ होगा. ठीक है, अगर वो कुछ proof हो जाता है, then it is a confirmed one. Okay, the mechanism is confirmed. Now in this case, okay, it is an alternate of the holiday model. Here, instead of having the two nick in the DNA strand, only a single DNA strand is getting nicked. Okay, and that strand is getting displaced and creating a holiday junction. अगर वो चीज clear है तो ये सब भी simple हो जाएगा. So you have got the DNA, okay, so अब homologous pairing हो गया है. So here you can see that only one is there because of the endonuclease, and this strand is going into the other one, and the process, the step is called the strand invasion, right? Now when you say something in ... उसका क्या मतलब होता है? That the DNA pol 1 is synthesizing the 1, okay? Now are you able to follow? See, this one is getting this one, nick ho gaya. This one is getting displaced. Displaced means the hydrogen bonds are broken, it is coming and it is attacking to the other one. Okay, ये जो dot दिख रहा है इसका मतलब DNA pol 1 आके वहां पे जो gap create हो रहा है उसको fill कर रहा है. Okay, next. So again, then the ligation is happening. So either you can have like this or like this, and ये आपका join हो जाएगा. See, if you see the ligation, यहाँ पे difference देखो, ठीक है? So again, this is forming a holiday junction over here, correct? Right? So if you have got the again the horizontal resolution, then you can see that I'm having the parental strand. Can you see? Okay.
Yes, ma'am.
Okay. Now here also, it just depends on how the branch is getting migrated. Again, if you see, I have here again the parental strand. Iska matlab, it was horizontal cleavage. If I don't see any parental strand, it means it was vertical. Simple. So this is how you need to understand the resolution. अब वो depend करता है कि उस time कैसे branch migrate हुआ है. So like in the video you have seen यहाँ से शुरू होके वो यहाँ तक गया. It's not necessary. वो वहाँ पे भी खत्म हो सकता था, बीच में खत्म हो सकता था, यहाँ end में आके खत्म हो सकता था. So it depends on that time like what proteins are involved and what are the conditions which is favoring the recombination. Okay. Simple. Now the other one, when you have a double strand break, so this holiday junction is again helping in repairing. A single double strand break is generated in one of the chromatids. इसका मतलब क्या? See, you have the homologous chromosomes. But abhi pehle kya tha, in the first scenario we saw that the break was here and the break was here, correct? Second mein humne kya dekha, only single strand break tha. Now what do we see is that in the same chromosome you have got the two breaks. Okay, now this needs to undergo the strand invasion. So what they're going to do is they make a gap over here. Now this gap means what? You had the endonuclease and exonuclease acting over here. So they have removed these certain nucleotides from here. Okay, gap bana diya hai. Okay, then it becomes easy for them to go for the strand invasion and results in the formation of the loop, forming the heteroduplex jo abhi aapne dekha. So you can see niche wala strand, if you see, it is able to go and do the strand invasion like this. So eventually this is going to go like this. So यहाँ पे क्या हुआ, आपका holiday junction बना. Clear? There are three differences are there. पहला, cut was over here, then the strand invasion migration was happening, resulting in the formation of the holiday junction. And depending on the horizontal and vertical plane, the cleavage of the DNA, you are having either the crossover or the non-crossover product. Second scenario, only one strand was having the nick. Okay. Third scenario, the same chromosome is having the two nicks. Okay. So this is like when you have a double strand break, double-stranded DNA break because of some damaging agents or whatever the UV radiations it can be. Okay. So again, this is how it is going to repair with the help of the homologous chromosome. Yeah. So you can see over here. So you can see that in this condition, you have got now two holiday junctions. Yes, you are going to have here two holiday junctions. जब एक था, so we had got those two scenarios. Now here we have got the two holiday junctions, so we are going to have the four scenarios. So if you just see, if you just follow over here, blue, yellow, blue, yellow. Okay, then this blue and yellow, blue, and then this entire thing. Either you can have this full, this blue, and then this entire thing. I'm just showing this one. Okay, the rest you can do yourself. Let's go one by one. Oh, this time here. Okay, so let's go one by one. So you can have this one, C, blue, D, okay, and the entire thing. So over here, either you can have, the other one is blue, sorry, blue, yellow, yellow, entire yellow. Are you guys able to follow me?
Yes, ma'am.
Huh? See, that's what they have written also. Either you can have cleavage at 2 and 3. So 2 and 3. यानी कि ये यहाँ से जुड़ जाएगा, okay? Or you can have cleavage at 2 and 4. So depending on these scenario, you can have different components. Okay? So you can have recombinant duplexes and non-recombinant duplexes. But you need to understand whether the cut is happening in the vertical plane or in the horizontal plane. अभी ऐसे में हमें समझ में नहीं आता. You just have to imagine. That's why I showed that video, like how the chromosomes are getting rotated. Okay, and then the vertical and the horizontal cut is happening. This image you can find in the Google also. Just go through once. I mean, no one is going to ask like how the cut is made and all. But please do understand, like whenever the double strand break is there and it is undergoing the repair, it's not necessary that even this is going to be there. Sirf ek bhi ho sakta hai, you can have two also. Okay, either you can have only one holiday junction form or you can have two also. So depending on the condition, like kitna aapka DNA damage hai, depending on that, it is going to have the holiday junctions and the repair accordingly. Clear, guys?
Yes, ma'am.
Okay.
Yes, ma'am.
I just wanted to take this much only.